Hi, everyone. Thank you for joining us. We're just going to wait a few more minutes to give some time for more attendees to join, and then we'll get started. Thank you all for joining our webinar today. Our webinar is going to be about 5 secrets to unlocking genetics precision diagnostics. We have 3 amazing speakers who will tell you a little bit about that. As I mentioned, we'll just give it another minute or two before we officially get started. I'm going to be your moderator for today. My name is Tamam Khalaf, and I am the genetic counselor that is based in the United Arab Emirates. Okay, I think we should get started. So thank you all for taking the time out of your day to join us and to attend this webinar. It's really an honor and a pleasure to have you all with us. We are part of Genomics. We are a diagnostics and reproductive genetics company. I'm Tamam Khalaf. I'm the genetic counselor based in the United Arab Emirates, and I'll be the moderator for today. I want to take the moment to thank our speakers, and I'll introduce them to you in a second, as well as our marketing team for putting this webinar together. So our first speaker is Dr. Lova Matsa. He is responsible for the implementation and the supervision of the genomics precision diagnostic services in the Middle East and India. He has over 12 years of experience in research, as well as 6 years of experience in the industry, with 15 publications in peer-reviewed national and international journals to his name. He focuses on the analysis and interpretation of the molecular genetic tests, including all the GPDX tests, as well as he also has explicit knowledge on the ACMG/AMP guidelines and next generation sequencing analysis and interpretation. In addition to that, he also supports clinicians as well as the genetic counselors in terms of test recommendations as well as clinical indications for testing. Our second speaker is Ms. Sara Al-Awesi. She's currently working as a genetic counselor in Genomics in Saudi Arabia. She previously worked as a genetic counselor in King Khalid University Hospital for 4 years. She also worked as a research associate in the genetic department in King Faisal Specialist Hospital and Research Center. She has a bachelor's degree in biotechnology from Egypt with honors. In addition to that, she also has a postgraduate master's degree in genetic counseling from Faisal University with first-class honors. She has more than 10 years of experience in genetics and cytogenetics, as well as molecular laboratory techniques. She's a member of the scientific committee in Genomics, as well as the Jester Association for Special Education, and she volunteers with support groups to offer them counseling in order to better understand their genetic conditions. And our final speaker is Ms. Ilona Barbosa. She is our experienced customer support manager. She has a demonstrated history of working in the hospital and healthcare industry, as well as a strong professional support system. And she has a bachelor's of science in pharmacy that's focused in pharmaceutical sciences from Southwestern University. So we'll be hearing from all 3 of our speakers today. If you have any questions, please feel free to type them in the chat box and we'll make sure we answer them at the end of the webinar. There's going to be an opportunity to ask questions at the end of the webinar as well. We'll be monitoring the chat box to make sure that we answer all the questions you ask. Thank you so much again for your time, and I'm going to hand it over to Lova.
Lova, please unmute yourself.
Sorry. Thank you, Tamam, for a wonderful introduction. And I would like to thank each and everyone for our monthly webinar series. And I would like to thank uh, each and everyone to taking time out of your busy schedule for attending this session. And in this webinar, we'll be discussing about 5 secrets to unlock genomic precision diagnosis with iGenomics. And we'll be discussing about these 5 most important points in this today's session, and how we are local and using the latest methods and the technologies for GPDX services, and increasing your exome accuracy to 99.4% at 20x coverage, and how the interaction happens through our genetic counselors and scientific support team with the clinicians and the patients. And we'll be discussing about how quickly we can deliver your results, the best possible turnaround time for the GPDX services we are offering. And finally, we'll be discussing about our customer service support to provide you 24/7 support. And coming to the first point, so we use the latest methodologies, and we are 100% local, operating everything from our Dubai laboratory. And genomic precision diagnosis offer a wide range of comprehensive testing methods using most advanced technologies for, for diagnosing genetic conditions at molecular level. And with the help of these technologies, we can identify sequence variants, structural rearrangements, trinucleotide repeats, and copy number variations. And with these broad spectrum of detecting these detecting the variations, so these uh, can be utilized in any of your life stages, right from preconception, where the family which is used for a family disease diagnosis, and couple who are planning to have a healthy progeny. So the couple can be tested for screening or the diagnostic purpose. Screening comes where there is no specific genetic condition in the family, but still the couple would like to go for a testing. To understand their uh, common the risk of having a kid, with the genetic condition. And we can also offer the couple whole exome for the diagnostic purpose, where there is an underlying genetic condition in the family, and we can able to identify the cause using our GPDX services and the whole exome sequencing. And we can also offer this testing for the prenatal diagnosis, where the ultrasound findings are compatible with a complex genetic syndromes, or if at all these adverse effects are leading to stillbirth, the product of conception can be tested using our GPDX services to identify what is the underlying condition. And we can also offer the test for the postnatal and neonatal. And we have a newborn genetic testing panel which covers all the inborn errors of metabolic disease genes. And the neonates or the newborns with genetic defects of any syndromic conditions or any developmental impairments can be tested for whole exome sequencing diagnostic. And we can also test any child or the adult with any specific genetic conditions, and could able to identify the underlying disease, genetic cause of the disease. And this is our full portfolio. So we have a wide variety of tests, right from the single gene test using MLPA, expansion repeats, and a full gene sequencing by NGS, and a target mutation testing using Sanger or Mini-6 sequencing. And we can also have an exome level or genome level testing for whole exome trio, couple, and solo, as well as we also have a chromosomal testing using chromosomal microarray platform. And we have 3 different platforms under chromosomal microarray, which we'll be discussing in later slides. And we have a precision panel. We have precision panels for all therapeutic areas like cardiology, metabolic diseases, and endocrinology, neurology, hematology, and osteology, so on. And as I discussed, we have a newborn genetic testing for the neonates, and we also offer this newborn for screening. So where there is a history of previous child affected with a genetic condition, and if there is a management available for such a condition, so we can offer this newborn screening to any neonate with a history of such a condition. And with the help of our certified counselors in-house and the geneticists, we offer support to the clinicians in choosing the appropriate test methodology to identify the underlying genetic cause. So we have chromosomal testings like karyotyping, FISH, QFPCR, chromosomal microarray. So karyotyping, we have at high resolution and regular karyotyping, and which targets all chromosomes. Aneuploidies can be detected by karyotyping, and as well as unbalanced translocations. So the balanced translocations can be detected by karyotyping, and the sensitivity of karyotyping to detect any chromosomal abnormalities is around 5 to 10 MB. FISH and QFPCR we can use to, this is mostly used for the prenatal services, where the most 5 chromosomes, 13, 18, 21, X and Y chromosomes, which are more prone to have aneuploidies, are tested using these methodologies. And yes, aneuploidies can be detected, balanced translocations, if any, known, can be detected using these, but yes, unbalanced translocations can be detected, while balanced translocations cannot be detected, and the sensitivity is around 100 kb. Chromosomal microarray, we have 3 different platforms: Optima, the low resolution 160K, 750K, the mid-range, and high density chromosomal microarray. And it's a genome-wide test, and aneuploidies and unbalanced translocations can be detected, while balanced translocations cannot be detected using CMA. And the sensitivity coming to Optima, around 1 MB losses and 2 MB gains can be detected, and 5 MB range of any loss of heterozygosity or absence of loss of homozygosity or absence of heterozygosity can be detected, and 750K, so deletions up to 50 kb and duplications up to 400 kb can be detected, while in HD, the deletions up to 25 kb and duplications up to 200 can be detected by high density chromosomal microarray. And other genetic testings that we offer, target variant testing, where a single variant is targeted per reaction, and which is done using Sanger sequencing, Mini-seq, or snapshot methods. And the purpose of this test is mostly for the carrier testing, where there is a known genetic condition in the known genetic variant in the family, and other family members would like to know the status of that particular variant, and those can be tested using this target mutation testing. And we also have full gene sequencing by NGS, and all coding regions of the gene are covered at a coverage of 150 using next generation sequencing methods. And when the clinician is sure about the diagnosis, and if that condition is known to have only one known disease causing gene, so the single gene approach can be utilized. Then we also have precision panels, where the genes associated with the condition or a special segment of the condition are grouped into a panel, and using next generation sequencing, we can offer these precision panel testing. And again, when the clinician is sure about the condition and we know certain genes are associated or certain pathways associated with the condition, so we can use this approach. And we also have whole exome sequencing for the diagnostic and screening purpose. And we cover all 24,000 genes, all protein coding genes are covered with exon intron boundary regions. Sorry. Yeah. We also have whole exome sequencing covering all 24,000 genes at a coverage of 150x using Illumina next generation sequencing method. And this is where a patient is suspected with a genetic condition and there is a combination of symptoms and the condition is like more syndromic, then whole exome approach is a better solution than going for a target panel or a single gene test. And we can also offer whole genome testing where entire genome is sequenced at 30x for the clinical purpose. And this is where when we don't have an answer using a whole exome sequencing or microarray, so still such a case can be taken forward for whole genome sequencing to identify if there is any novel gene that is associated with the disease condition. And we also have a single gene test specific to any condition by using MLPA and expansion repeat testing. And MLPA is a test where which is used to detect a deletion duplication at the genome at a single gene level. So we know that certain conditions like DMD, Duchenne muscular dystrophy, spinomuscular atrophy, and congenital adrenal hyperplasia (CAH) uh, uh, are caused by genes and where the deletions and duplications are more implicated than sequence variants. And these conditions can be first tested by using MLPA to identify the disease-causing deletion, and expansion repeat testing is done by a PCR technique and to identify these dynamic mutations, the trinucleotide and tetranucleotide repeat expansions, and the conditions known for associated with these trinucleotide repeat expansions or tetranucleotide expansions are like spinocerebellar ataxia, Huntington's disease, Fragile X. So these conditions can be tested using expansion repeat testing. And this is the NovaSeq 6000 instrument that we are using for whole exome sequencing purpose. And this is the system most powerful instrument performing whole genome level sequencing and more cost effective. And we also have Sanger sequencing and other instruments which can help us produce the data in time. And we'll be talking about how accurate and how our results are using these latest platforms. And the latest version of Illumina, that is NovaSeq 6000, is used for whole exome sequencing. And the panel that we are using from Illumina exome panel, and which is enriched with more probes for copy number variation detection as well. And we have in-house bioinformatic pipelines, and we can able to detect copy number variations and mitochondrial genome using our panels. And the exome panel from Illumina is of 45 MB in size, and the mean coverage is around 150x that we are targeting. And more than 95% of the reads are qualified for the analysis. That is, this data is at Q30, and 99.4 and 99.3% of the data around at 20x coverage are like covered of ClinVar and HGMD disease causing mutations. And more than 90, the sensitivity of detecting copy number variations using our NGS methodology is around 90%. It is around 90% when it comes to a homozygous deletion or duplication. And the accuracy is greater than 75% in general, and the accuracy and sensitivity of copy number variations by NGS is always depends on the size of the copy number variation we are detecting and the zygosity. And This is our in-house analysis viewer, how we analyze the data using the in-house viewer that we have. And we use even the in-house bioinformatic pipelines tools and maintaining the confidentiality of your patient information. And we do not need to outsource or upload the data onto third-party software, so that since we have everything in-house, and we follow ACMG guidelines to classify these sequencing variants. And we have certified geneticists and genome analysts in-house to perform these curation and interpretation of the data. And these are some databases that we use in our pipeline and the interpretation of the data, like Nomad, OMEM, HGMD, the paid version of HGMD, CleanVar, CleanGen, and some other databases. And overall, talking about our bioinformatic pipeline right from the sample collection. So we have, once the sample is received to our lab, the sequencing, DNA extraction for the sequencing, and then library preparation and sequencing using the instrument that we have, and bio IT pipelines, and the curation of interpretation of the data using different databases and in-house pipelines that we have. And finally, putting all this information into a comprehensive reporting format where a clinician and patient can understand what the disease and the underlying cause for the disease is. And we have scientific support throughout the process, pre and post test. And especially once the report is released to the clinician and the patient, upon a request, we can always arrange a call with our counselors to have a detailed discussion about the results and further what steps can be taken to manage the condition in the family. And here I would like to introduce Ms. Sarah, who is our qualified counselor, and she will explain more about how we interact with the clinicians and patients, and discussing about the results and pre and post test counseling sessions. Welcome, Sarah.
Thank you, Dr. Lova, for this informative lecture. My name is Sarah Louisi. I'm a genetic counselor based in Saudi Arabia. And what makes Genomics unique, that we offer the genetic counseling services free of charge for all of our patients. We offer the genetic counseling for doctors and patients at the same time. To give you more information about genetic counseling will give you more information about how genetic condition might affect the individual and his family. You may think to refer your patient to a genetic counselor if you have a patient who have a sign and symptoms of the disease that might be a genetic. Sometimes if you have an individual with a strong positive family history of one of the genetic condition that makes them more likely to have the condition, you can think of referring this patient for genetic counseling. If you have also a couple who are identified to be a carrier through premarital screening, and also if you have a pregnant woman who's worried and would like to know her risk for having an affected child, especially with chromosomal aneuploidy, or sometimes we can also support the woman during the pregnancy if she had abnormal test result, or if she had a previous child who diagnosed with one of the genetic condition. You might think to refer the patient so in order to benefit from genetic counseling. As I mentioned, that we offer pre-test and post-test counseling for both doctors and patients. We help doctors to understand the interpretation of the genetic report. We help them to understand what does that mean and the implication of the genetic test result for patient and the family as whole. We also do risk assessment, and we do the counseling on phone or in person, or sometimes we do the conference call. So in terms of counseling, we offer both counseling and education for both doctors and patients. For pre-test and post-test counseling, in the pre-test, we usually discuss the suspicious diagnosis, why the patient is referring, and the suspicious diagnosis. We discuss the signs and symptoms of the disease. We discuss the inheritance, the recommended genetic testing, and the turnaround time. For post-test counseling, we discuss with the patient the confirmed diagnosis. We discuss the genetic test result, if she has this positive result, or sometimes if she has an inconclusive result. We offer the extended family services in terms of counseling other family members who are at high risk to be a carrier of the same condition. We can offer the future preventive options to avoid the recurrence in the future pregnancy. We make sure always that the patient chooses the test. He is fully aware about the test and the limitation of the test, and he chooses to pursue the testing without any pressure. And we do also the recommendation once we do the test, and we can send the counseling note to the patient and to the referral doctor as well. In terms of education, we are providing the information for doctors. We are raising the awareness for doctors who are not familiar in genetics. We provide depth knowledge about the test description, including the indication and the application of all of our genetic testing. We do a lot of like workshops and meetings. We do a series of webinars like this, and we participate in conferences, and we have our unique educational hub where patients can access the website, and they can read more about the genetic services that we provide. In terms of supporting physicians, we are helping the physicians who are not experts in the field to choose the best test for their patients. We make sure that what they have ordered is the right test for their patient. We can help them to understand the genetic report, and we do the molecular confirmation in case we have identified a variant in the initial report. We do like a family segregation, and we do another genetic testing if it's needed. At the end, we consider that we provide a future preventive option for the patient to help them to avoid the recurrence, either by providing the prenatal testing during the pregnancy or offering the pre-implantation genetic diagnosis for this family. We have a local team to support all the doctors during requesting genetic testing. Our main advisor, Dr. Ayman Al-Hattab, he's a scientist and geneticist based in UAE. Me and my colleague Tamam, we are a genetic counselor, and we are offering the genetic counseling services for all patients in Middle East. And our lab director, Dr. Lova. We also have an international team who are always available for additional support when it's needed. Again, we are providing the genetic counseling for all patients free of charge. and we support the genetic counseling in all life stages. Thank you so much, and I think now I welcome the next speaker, Miss Ilona.
Thank you, Sarah, and thank you, Dr. Lova, for that edifying presentation. Good evening, everyone. My name is Ilona, and I am part of the Customer Concierge Team, 24/7 Customer Concierge Team. Genomics offers global support, having 22 laboratories, still expanding with a solid and strong team to provide the best assistance and supports to clinics and doctors. This time we will discuss how we handle and manage the supports locally. We have 7 easy steps. First, we have a genetic counseling session in which after the counseling session done by our genetic counselors to clinics, doctors, or even patients, they will notify us and give us any information that we need now to contact the clinics or doctors and patients in order for us to educate or to inform them the proper way, the step-by-step process on how to handle samples and as well as how to fill out the forms. Once the samples has been collected properly, then it will be shipped to iGenomics, in which we will tackle later on. It will be shipped to iGenomics with couriers or our domestic drivers. Then once received, we will do the testings, analysis, then we will proceed to report generations, and for the last steps, to release the result within a specific given time. As mentioned here, it will be 15 working days turnaround time. Here, we have different types of samples as well as the volume requirement, transportation temperature, and the container to use for each specific sample type. For blood, for referral blood, we have 3 ml of volume requirement that can be transported within 20 to 25 degrees Celsius. For purified genomic DNA, it should be kept or sealed in an Eppendorf tubes and can be transported in a room temperature, having a minimum of 1 microgram of DNA at a concentrations of 50 to 100 nanogram per microliter. Then we also have product of conception or fetal tissue, which you can put or you must put in a sterile container, wash it with sterile saline solution, and then ship it within 20 to 25 degrees Celsius with a volume of 3 to 4 millimeter product of conception specimen or 50 to 100 milligram of each tissue. Then we have amniotic fluid and chorionic villi. For amniotic fluid, it is a must that we should put it in a sterile container, transport it within 20 to 25 degrees Celsius within 10 to 15 ml volume. And for chorionic villi, you have to also put it in a sterile container with a culture medium or saline solution with 1% antibiotic. Then it can be transported between 2 to 8 degrees Celsius with a minimum volume of 300 to 500 milligram. Having said that, I would just like to remark as well that here in UAE, the temperature is quite high, so we also recommend for the samples to be shipped along with a cooling pack or gel packs, just to avoid bad quality of the sample upon receipt in iGenomics lab. Here are some examples of the kits that we are using. First, this is the tubes that we are using for the blood, EDTA vacutainer tubes. And then we also use some sterile container with a red cup. This is the one that we use for product of conceptions. And for amniotic fluid and chorionic villi, we are using conical tube, which you can identify it with a white purple cup. And Anyway, all of these kits will be provided to each clinics and doctors and patients who will be requiring the kits for their test. Now we are launching and having 15 working days turnaround time. We are speeding the process because recently we have launched in-house testings, and this allows us to provide the best possible turnaround time for each service. For 15 working days that we have whole exome diagnostic, whole exome screening couple, whole exome screening customized panels, CMA or chromosomal microarray, single gene, target mutations, and as well as MLPA. And for 20 working days turnaround time, we offer it for whole exome trio and whole genome sequencing. For us to properly track the samples and your samples upon receipt in iGenomics, we are using our Navision software that will help us to track and make sure and ensure that the sample will be received on time upon we receive it from iGenomics laboratory till the result will be released. Microsoft Navision software that we are using, we can identify the patient names, the medical record number of the patient, as well as the deadline and the deadline warning, and to ensure that the samples will be released within a specific turnaround time. iGenomics customer support are divided into two groups. We have sample management team and as well as the logistics team. For sample management team, we ensure that upon receipt of the sample, we validate the forms and the samples identifier to ensure that the samples are fit with the forms that has been given to us. Then we forward it to our laboratory. Then for logistics, it's a team that track day-to-day basis of all the shipments that has been collected from your facility until it reach iGenomics lab. We are one department dedicated to unify and manage everything during the testing cycle. We are 24 hours, 7 days a week available for any critical situations and urgent matters. We are also using multi-channel contact options such as WhatsApp, website, and emails. You can also communicate or connect with us so that we can assist you to connect with any different departments in our laboratory. We can connect you to finance, marketing, and scientific teams. Also, we follow up and we ensure that doctors and patients receive the reports on time. Again, we are the one who checks and validate documents, informations, tracking, and releasing the reports as per given turnaround time. Logistics teams in iGenomics are the ones who deals with the daily tracking of each shipments. We are the ones who dispatch the kits to the clinic and to collect samples from the clinic. We also do coordinating such shipments, international and domestics. For internationals, we use professional couriers such as FedEx, DHL, Aramex, UPS, and for domestics, we have qualified drivers, well-trained drivers. Also, they are fully vaccinated, and they ensure that the samples is properly taken care of from the time it has been collected from your facility till it reach iGenomics lab. Let me introduce to you to our team here in UAE, which you can contact. First, we have Romella, Rachel, Daniel, and yours truly. Also, we have our team in Kuwait, which is Mr. Ali, and in Saudi Arabia, Mr. Adam and Mr. Youssef. Also, you will be able to contact us through different email addresses as well as telephone numbers which has been provided. For UAE, we are accepting calls through toll-free telephone numbers and mobile numbers. And in Kuwait, we also have a mobile number and with the email address, same as with Saudi Arabia. So these are the contact information or contact details that you can reach if you need some assistance for any collections from your facility. Again, we would like to say thank you for allotting your time to attend this webinar and hope everything is clear. And if you have some questions, you will feel free to send us questions through chat box. Thank you.
Thank you, Sarah, Elona, and Lova for a wonderful presentation. We thank you so much for your time. So we have a few questions just before we close up the webinar. I think the first one's for you, Lova. How are ACMG secondary findings reported in the whole exome sequencing? If you could just give us a little bit more information.
Yes, sure, Tamam. So we can report ACMG secondary findings through whole exome sequencing, and upon it, like we have a special column in our test requisition form. So upon a consent from the patient and the clinician, yes, we can able to report these ACMG findings, all the pathogenic and likely pathogenic variants covering in these 83 ACMG genes recently identified. And we can put a separate section in the report itself saying these are ACMG secondary findings, and a special a special phenotypic evaluation is recommended for those reported variants.
Definitely, thank you for clarifying. And I also would want to say that we would definitely recommend counseling for identification of the ACMG secondary findings so that the patients are well aware of what information they could potentially receive. Coming on to our next question, Sarah, you mentioned genetic counseling is free of charge. How can we access this? One of the doctors is asking, how can they access it and their patients access it?
Yes, Tamam, we offer the pre and post-test counseling free of charge for all doctors and patients. For doctor, they can just call us if they would like to have a verbal counseling, or sometimes they can refer their patients to be counseled by us. And after that, we can send the counseling note to the referral doctor. For patients, sometimes they can book their genetic counseling session through our website. They can go and just book an appointment, or they can send us an email, or they can physically come to our lab in Saudi or maybe in UAE to meet a genetic counselor before proceeding with any genetic testing.
Wonderful. Thank you for that clarification. And just a quick last question. Lova, you mentioned the different types of testing, and if we did the whole exome sequencing test, which is quite a comprehensive test, why would we need to do other types of testing as well, like MLPA?
Yeah, so as we know, each and every technology has its own limitations. So the same for whole exome or the NGS-based technologies. So though we have like copy number variation detection methodology through NGS, so still there are like to have 100% confidence on the deletion on duplications of a particular gene. So an alternative method like an MLPA is used to rule out or to identify any deletions or duplications in that particular gene. Or like sometimes we can go for chromosomal microarray. So if there are any small deletions or duplications or any copy number variations that are causing the disease, which definitely can be skipped by NGS or the CNV detection methodology. And yes, of course, there is another like a separate testing for the trinucleotide repeat expansions as well.
Okay, and just to confirm, you would be able to support the doctor and the patient in choosing the most appropriate technology depending on the clinical symptoms that are present.
Yes, of course. As we were mentioning, we have like free pre and post test counseling sessions. So which are like, as Sarah was mentioning, yes, the doctor or a patient can approach us through emails or phone numbers that we are providing here, and they can send us a case, any case like with the clinical notes or the observations that they have for the case, and accordingly we can choose or like help them choosing a right and appropriate technology for identifying those mutations.
Okay, thank you so much. Well, we're coming to the end of our webinar. If you have any more questions, this is the email that you can use, info.me@genomics.com, and we'll make sure to cover any questions that are remaining. Thank you so much for your time, and I want to thank the speakers again, Lova, Sarah, and Ilona, as well as our marketing team for setting all of this together. Thank you all for your hard work, and have a good night, everyone.